Hey guysies, this is going to be our blah blah blah. Let me back up. This is going to be our part two lecture on male anatomy and physiology. I want to back up because I was going to skip this part, but I feel like it's necessary. Sperm anatomy. So, I don't know if you learned this in med term or your other health classes, but sperm has its own little structures to it. And we talked about the major male structures, anatomical structures in the last lecture. But I want to really zoom in on sperm here. And we're also going to dive into something called spermatogenesis. And you've probably learned this before in your other classes like biology or even seventh grade science, because I taught it last year to my kids. And that's going to be more physiology. So we probably won't go too much in depth on physiology as far as talking about all the nerves that work on the male and everything like that, because we don't have time. But I do want to mention this topic right here. So what is spermatogenesis? It's essentially just making sperm. Okay, so if you recall, where does this happen at? It's going to be in the testicles, and I'm going to remind you of where exactly in the testicles this process is going to happen. So gametes. So we use this word, we use gametes, we use gonads, we use those words all the freaking time, but what do they mean? So gametes are going to be like the sperm and the egg, all right? Gonads are like the testicles and the ovaries, okay? So they're the actual like structures that we think of. But gametes are definitely the sperm and the egg, or the ovum. So this is the process of creating sperm. In women, it's called oogenesis, and we're going to talk about that on Monday and Tuesday. So you've probably heard of this term before. It's pretty basic. It's called haploid, and that just means making something that's doubled into half the number. I remember it because half starts with an H, and haploid starts with an H. When it's doubled, it's called diploid. And so I remember double diploid. And you can see here that meiosis its whole point is to make something that's doubled into half. And you can see here we start with 4 chromosomes, and then eventually we're going to end up with half of 4 in each of these guys right here. And we're going to talk about how meiosis applies to the spermatogenesis and what that means exactly. So where does this happen? So it happens in the testicles, the testes. Again, if we were to cut it in half, which I don't recommend, it has these wavy structures called seminiferous tubules. And this is kind of, like we said, the sperm factories. It's going to make sperm that are eventually going to be stored until they're completely mature in the epididymis. And then they're going to be shot into the vas or ductus deferens when they're mature and able to swim. So here is spermatogenesis in a nutshell. So spermatogonium is just the stem cell for sperm. Okay, so we talked about stem cells. I always think of spermatogonium like a little flower. I don't know why I think of it, but they're flowering sperm, and they're just the stem cell. And these guys are going to be diploid, so they're going to be doubled. That's what 2N means. It means they're diploid. So the whole point of spermatogenesis is to get the sperm, or the sperm, to be haploid. Okay, so half of the chromosomes from the doubled number here. So spermatogonium are going to differentiate. They're going to go through mitosis. So here's mitosis right here. You learned this in like 8th grade or 7th grade probably. So the spermatogonium, they are going to split by mitosis. And mitosis, it's going to create the exact same thing. Just like whenever you scrape your skin, mitosis is going to make new skin cells that are identical to what they were. But the whole point is just to get more of them made. And that's what's happening exactly right here. So when the spermatogonium goes through mitosis, it's going to become a primary spermatocyte at that point. And we're still doubled. Okay, we're still diploid here. So primary spermatocytes are going to differentiate and divide even more, and they're going to create a secondary spermatocyte. And this is when meiosis 1 or meiosis 1 is going to kick in. Again, we're not doing mitosis anymore. We're going to do meiosis. This is the whole process in reproduction right here, not mitosis. Okay? And at this point, between the primary spermatocyte and the secondary, we're going from diploid, so double the number, to half the number at this point. The secondary spermatocyte right here is going to go through another round of division through meiosis 2. And that's going to create something called spermatids. And they're all haploid. There's 4 of them. And you can see here that there's 4 sperm or spermatozoa. So I might ask you on the test, how many sperm are made in spermatogenesis? And you would say 4. It's always that number. And the whole point of it is just to get more made. And so we want them to be haploid because when they join with the ovum, the female gamete, she's going to be haploid too. So when they meet up together, they're going to become diploid. So you and me, we are diploid organisms. So that's the point of having it. So where was I? So over time, these spermatids are going to have time to mature. They're going to be nourished. They're going to be fed. They're going to be supported in the seminiferous tubules. And they are going to eventually be sent to the epididymis for maturation and then sent out into the vas deferens. So here's some basic sperm anatomy I want you to know. There's not too many things, but I do want you to be aware of the nucleus. And this is where the genome, the genes, the chromosomes, all that good stuff is stored. And I think that's pretty obvious. So everything's going to be stored here. So when the sperm penetrates the female gamete, then their haploid genes are going to be joined with theirs, and it's going to become a diploid organism. So the next structure is the acrosome. It's this little pocket right here, and it contains enzymes that allow the sperm to penetrate the wall of the egg. So if we didn't have that, then it would be like the sperm's just hitting a brick wall. It wouldn't be able to penetrate and then give its genetic material to the female. And the flagellum is for locomotion. You know what a flagella is used for. If you didn't have flagella, then it wouldn't be able to move anywhere. Okay, so more about the seminiferous tubules here. So again, this is in the testicle. It has these cells, and this will definitely be on the test when the time comes. They're called serotoli cells. And their whole purpose is to nourish and support the precursors to sperm until they're ready to be sent to the epididymis for further maturation. So it's kind of like when you're at your parents, well, you're like the spermatogonium. They're making you mature, they're nourishing you, they're supporting you. You're sent off to college and you're grown up, so you're these guys right here, but you're still needing some support. You're still needing them to give you their credit card number for McDonald's and Arby's. But over time, you're going to be completely out on your own. And it's going to be a race for who's trying to get the diploma. Okay? So eventually these guys are going to be sent to the epididymis, and that's going to be kind of like your college experience. And then the epididymis is going to shoot you out too, and you're in the real world, baby. So serotoli cells nourish and support the precursors. And we talked about the precursors are the spermatogonium, the primary spermatocyte, which we see here, and then the secondary spermatocyte, which I'm assuming that's what this guy represents. Okay, and then the spermatids, which is there, and these are the sperm that are trying to mature. Okay, so I think that's it. Thank you, and let me know if you have any questions. Make sure you're writing this down.